Good afternoon, everyone, and welcome to our University Research Week session on stress and our mental health. I'm Troy Blackburn, Associate Dean for Undergraduate Research, and along with my colleague, Dr. Bob Pleasance, it's my pleasure to moderate today's session. The COVID pandemic and other recent challenges have introduced all of us to severe stress. This session will feature experts from UNC-Chapel Hill who will talk about the impact of pandemic stress on mental health and how current research here at Carolina is driving innovation in neuroscience that leads to new treatments and better outcomes, as well as approaches for effective stress management. Our first speaker will be Dr. Samantha Meltzer-Brody, who is the Assad Meymandi Distinguished Professor and Chair in the Department of Psychiatry and the Director of the Center for Women's Mood Disorders. She will provide an overview of the impact of COVID on mental health, including anxiety and depression. She will also discuss a novel drug developed for treating depression, for which all phases of clinical trials were conducted here at Carolina. Our second speaker will be Dr. Jose Rodriguez-Romaguera, who is an assistant professor in the Department of Psychiatry and co-director of the Carolina Stress Initiative. He will discuss the underlying neuroscience of stress using animal models to highlight how stress impacts the nervous system. Our final speaker will be Dr. Crystal Schiller, who is an assistant professor in the Department of Psychiatry and co-director of the Perinatal Psychiatry Program and director of its Psychology Internship Program. At the conclusion of the three talks, we'll have time for question and answer using the Zoom chat function. Please enter your questions in the chat and we'll convey them to the presenters. Without further ado, please join me in welcoming Dr. Samantha Meltzer-Brody.
Thank you. I'm delighted to be here and welcome to our session today. We're going to be talking about stress and our mental health and looking at this through multiple different lenses. And I'm thrilled to have my terrific colleagues in the Department of Psychiatry here with me today. I want to talk about the scope of the national mental health crisis. The pandemic that we've been living through, as we all know, has markedly worsened the mental health of the United States. And this is new data from Mental Health America 2022 report. We now know that 1 in 5 Americans, 46.6 million people, are suffering from some sort of mental illness. That may be anxiety, that may be depression, that could be an eating disorder, that could be a psychotic disorder, that could be any number of things. And what has happened is that the current infrastructure in this country to address mental health issues is inadequate to address the massive need. And about 25% of adults who have some sort of mental illness are reporting an unmet need for treatment. So these are really devastating numbers. And in the Department of Psychiatry, and we are part of the School of Medicine and work with UNC Health, we have been running as fast as we can to try and keep up with this massive demand and massive need that we face now. So as you can imagine, if the prevalence goes up 3 to 4 times of anxiety and depression, you don't automatically have the workforce able to tackle this go up 3 to 4 times. In youth and in high school and college-age people and beyond, we have seen marked worsening. I would say that the mental health situation of our youth is fairly dire right now. The mental health concerns in this age group have more than tripled in the last year. So now you're looking at, over 10% with a severe major depression, 15% with some sort of major depressive episode and beyond. And we certainly know this is associated with all kinds of morbidity and unfortunately increased rates of suicide. And so this is something we're taking extremely seriously. Just to give a little more information about the scope of the national health crisis. For families with kids under the age of 18, the demands of caretaking during the pandemic have been substantial, huge impact on mothers and kids, but in teens and young adults, it's been particularly cruel. And so if you look at CDC data, mental health emergencies rose by 24% for kids between ages 5 and 11. These are for mental health emergencies, and by 31% for children and teens 12 to 17 years old. Communities of color have been disproportionately impacted. 48% of non-Hispanic Black adults and 41% of non-Hispanic white adults report symptoms of anxiety or depressive disorders. And this is all going to require a massive and new effort, new innovative ways to address these really huge, complex mental health issues, and will take all of us working together with new funding and resources and new ideas. So I'm hoping to those of you listening today that some of what we can do is help get your new ideas and convince you why research in neuroscience and psychiatry and behavioral health is really an incredibly exciting and important time. My work has really been focused on translational approaches, and I just want to have a nod to translational research, where you take basic science that can be preclinical or animal studies that define mechanisms, targets, and lead molecules, and then translate into humans with this series of clinical trials from phase 1 to 2 and 3, ultimately translating into practice that then can lead to community impact, population level health outcomes that can be powerful. And so this has really been the focus of my work of over 20 years at UNC, a big focus of the UNC Department of Psychiatry, and you're going to be hearing some more about this today. My work has been focused on maternal mental health, and the impact of COVID on maternal mental health has been terrible. As you can imagine, the perinatal period is vulnerable. The social isolation, the increased stress has been extraordinarily difficult for women and for mothers. And so I think this is really a call to arms, and a lot of the work that we've been doing has been looking at how we translate science and the benefits for the clinical benefits, the community, the economic, and the policy benefits. I'm going to be touching on all of those today. There has been a great need for new and effective and rapidly acting therapies for postpartum depression. We know the postpartum period is a critical time for maternal-infant bonding, and that negative effects on the mother or offspring can be long-lasting when they go untreated. So this has been an area of research, and I'm using this as an example. You could apply this really to any area of mental health on how you can take an unmet need and a public health need and turn it into a really robust area of research that can have both clinical as well as policy implications. So one thing we've been looking at is for years is how can we increase the pace that we're able to successfully deliver treatment. And so how can we use, in the case of women with severe major depression in the postpartum period who may be suicidal, how can we deliver treatments that more rapidly act? This is a critical window. How can we improve interactions for mom and baby and reduce morbidity and mortality? So we know that postpartum depression, the prevalence is 10 to 15% in the general population, and the underlying cause is unknown, but likely multifactorial, has been linked to all kinds of biologic processes, inflammatory signaling, fluctuations in perinatal hormones, dysregulation of stress pathways, including the HPA axis, GABA signaling, et cetera. We know it's more heritable than non-perinatal depression, with 44 to 54% in perinatal versus only 32% outside of the perinatal period. And there are multiple hypotheses about why some women get postpartum depression and others do not. These can include endocrine mechanisms. We know that during pregnancy in all women, normal physiology is levels of estrogen and progesterone go up very high and then plummet at the time of childbirth. That's in the top left-hand corner, at least how I'm looking at the screen. There's genetic and epigenetic mechanisms looking for an underlying genetic signature or epigenetic changes that may be associated with risk, synaptic transmission mechanisms, neural network mechanisms, inflammatory mechanisms, neurosteroid mechanisms, including allopregnanolone, which is the neuroactive metabolite of progesterone, and stress-related mechanisms, dysfunction of the HPA axis. So all of these are candidates. There's literature in all of them. And likely with most things in science, it's not going to be any one thing. It's going to be specific to different women based on their history, their underlying biology, and their life events. So here's an example of what we've worked on, and everything I'm going to present today represents team science. I just want to highlight that with translational work and the work I've done, this all represents having team science with wonderful colleagues who each bring multiple different skills to the table. And it's one, great fun, and two, no one person is going to be able to do any of this themselves. So really collaborating with people is one of the best things that I enjoy about being at Carolina. We developed a app, the initially called the PPD ACT app, the Postpartum Depression Action app, to understand the genetic signature of postpartum depression. And this was something that we worked with the Apple Research Kit, launched in 2016, to use the research kit to bring science to anyone who had a smartphone. This was the first study that used an app to ask people to contribute genetic samples, and was very, very successful in landing a big splash. By 2019, we had been reached out to because of this by a PR company in LA that said we had won a competition, not that we knew we were in a competition, for a pro bono investment. And they essentially said to us, we think that your PPD ACT app is cool, but we think your branding is really JV. And we're going to give you $1 million in pro bono funding to help rebrand it as Mom Jeans. This launched an entirely sort of new program, and we've been getting DNA samples through this, rebranded as Mom Jeans. That's been really fun and exciting, and leading to what is now going to be the largest genetic study of postpartum depression for a genome-wide association study that's being written up now. And why is that important? Well, that's going to help us understand, who is at risk, and ultimately what treatments are going to be most effective. So I'm going to switch gears from that and talk about brexanolone. Brexanolone is a proprietary formulation of allopregnanolone. And you may remember that I mentioned that allopregnanolone is the neuroactive metabolite of progesterone, a female gonadal steroid that goes up along with estrogen normally in all women that have postpartum, that are pregnant. What's interesting is that there'd been preclinical work and lots of reasons that people suspected that allopregnanolone might be useful in treating postpartum depression, but really a very tricky compound. It's a positive allosteric modulator of GABA A receptors and impacts both the extrasynaptic and intrasynaptic receptors. And so we were approached to give to participate in what would be the first open label study, the first person ever infused with this proprietary formulation. And fast forward from the open label study of 4 patients that we did at UNC in our perinatal psychiatry inpatient unit, which was the first and remains one of the few in the country, which is really exciting, to double-blind phase 3 studies, and led to FDA approval of the first drug for postpartum depression. The timeline was rapid. It was exciting, and this is sort of the first patients done and the papers published, leading to FDA approval in March of 2019. And interestingly, within 2 weeks of March of 2019, it was the second drug right after intranasal ketamine to be approved after a very long period of time, like decades before sort of a new form of an antidepressant and a fast-acting form of an antidepressant, fast-acting, was approved. So exciting time in the field. I'm not going to spend a lot of time going through the studies. These were published in the Lancet paper, but basically what's novel, this is an IV drug, a 60-hour infusion. People have been followed out 30 days. The active drug was clinically and significantly superior to the placebo, persisted at the 30-day mark. The side effects, the primarily one was sedation, and that's something that has to be carefully monitored. But this is a new paper from our team, with Dr. Raya Patterson being the first author, that we are now a national referral center for people coming in to give brexanolone on our inpatient perinatal psychiatry unit. So it's been approved now for a couple years. And despite the demands of the pandemic, we still give it. And it really remains a huge blast to see people have a marked response with a fast-acting antidepressant, particularly for women severely ill, many of whom are not able to function or who have suicidal ideation. And lastly, I'll end with this. This work that we do, and a big focus of the UNC Center for Women's Mood Disorders, is also focused on policy. And that's something that Dr. Schiller, you'll hear more from. But this is a paper recently that just came out in Health Affairs on how we look at policies and programs. So, for example, why is UNC still one of the only inpatient perinatal psychiatry units in the country? Why is that not something that's developed regionally? Why is it so hard for some people to access care? How do we make sure we have adequate resources and funding to take care of all mothers during the perinatal period? So to conclude, translational research that bridges bench to bedside offers the greatest promise to improve outcomes for our patients. I've given you the example of the rapid antidepressant effects of brexanolone as a positive allosteric modulator of GABA and a fast-acting antidepressant. And this is an example of using one of the underlying hypotheses of the pathophysiology of postpartum depression to develop a novel treatment, and became the first drug approved for PPD. How we then apply that so you can impact the community and disseminate broadly to ensure the broadest public health impact is then a whole separate phase of translational work, and one that can be really, really meaningful, but does involve policy and figuring out how you get beyond barriers. So I just want to thank again all of my colleagues with the UNC Center for Women's Mood Disorders. This is a huge team effort of many, many people. You'll see many names here. I won't go through all of them, but this is something that is really reflects, I think, the collaboration and collegiality of UNC and why it has been just such an honor and privilege to be here for 20 years. So I'm going to now end with this and invite my fantastic colleague, Dr. Jose Rodriguez-Romaguera, who's going to change gears considerably and talk to you from a basic scientist point of view. Stop sharing.
Jose, you're muted.
Can you hear me now?
Now we can. Welcome.
Perfect. Can you see my slides? No.
The slides went away. We saw them and then they went away. So you have to bring them back up here.
Doing that right now. All right, can you see them now?
Yes.
All right,
Success.
perfect.
All
Thank you,
right.
Dr. Meltzer-Brody, for that introduction, and thank you for all the wonderful work that you do here in our Department of Psychiatry. So as Dr. Meltzer-Brody mentioned, I'm going to be switching gears a little bit and talking about how we can use animal models to study the neurocircuitry of stress and anxiety. So one of the things I really want to have everybody think with me of how we think about the brain nowadays. One of the things we think about the brain is like brain circuits, how really you have different brain regions that are really talking to each other constantly throughout all the days of our lives. And with that, I really want to talk about how these different brain circuits are talking to each other. So we have neurons in the brain, neurons that send projections to one brain region or to another, and are really transmitting information as we process the environment around us. But one particular thing also about like the brain, and in particular the human brain, is that there is some homology in terms of how the brain is organized across species. So for example, the mouse brain shares many brain regions and circuits that are also available in the human brain. So we can use mouse models to really probe into the brain and really understand basic circuitry. In my lab in particular, one of the things we study is the bed nucleus of the stria terminalis, which has been recently talked about like one of the brain regions involved in anxiety and stress related behavior. So with that, one of the things I like to do during my talk is to really take a magnifying glass and look at the BNST and how it encodes anxiety and stress related behavior. With that, one of the things I like to talk about that we can do in basic science is we can use all the techniques that we have available to us to really hone in into brain regions such as the BNST. So we can use microscopy techniques, for example, to look at different cell types within the BNST, to look at different neurons and really analyze at a single neuron level, how are they encoding stress and anxiety related behavior. One of the things my lab focuses on is studying specifically these prepronoceceptive neurons, or as I will call from now on, PNOC neurons. And as you can see here in this image, that we have in blue, we have all neurons within this particular brain region, but in green in particular, we're only labeling the neurons that have this PNOC gene. And one of the things we're really interested in is studying how these neurons encode anxiety and stress related behavior. So with that, one of the things I really like to highlight is in an anxiety disorder. So one of the things that really is particular about them is heightened arousal responses, one of the key symptoms. So you could think about like, how can we study heightened arousal responses in a mouse model? Both species actually have arousal responses, so we can use that as a way to really target how neurons, like for example, the pre-pronoceceptor neurons, encode states of arousal. And with that, we can design an experiment where we put an animal under a 2-photon microscope to really visualize the activity of these neurons. And one of the things that we can do to induce an arousal response is we can expose them to certain odors that are relevant to their and anxiety inducing to them in particular. So for example, we can expose them to something like fox odor. So we can visualize what the neural response is happening to fox odor when they smell fox odor, where fox odor comes near them. And then also in parallel, we can put cameras and look at the eye as a way to measure arousal responses. So like how the pupil, for example, responds to these odorants that are inducing of like an arousal response. And with that, I really want to bring you into the lab for a second and really show you how one of those experiments looks like. So here we have a head fixed behavior setup where we have an animal, as you can see here on the left in a running wheel under a 2-photon microscope. We bring forward an odor that comes from fox odor, and then we look at the neural responses and the arousal responses that is happening while the animal is exposed to this odor. So first I'd like to show you, here's what we can visualize on the left, where you can see these little circles that are lighting up. Those are actual neurons that are lighting up. And we can actually extract the signal of those individual neurons so we can study how their patterns correlate with arousal levels. So here what I'm going to show you is recording at the same time as an arousal response. So what you see on the left is actually the pupil of a mouse. And when you see on the right is the data of like the pupil size and 2 example neurons. And you'll see what happens when TMT, the predator odor, or the fox odor comes on board. So what you can start seeing is that the predator odor is increasing the size of the pupil, sort of that arousal response that I was talking to about earlier. But we have 2 neurons here, one that's getting excited the same way as that arousal response from the pupil, and another one that's actually going in the opposite direction, getting inhibited. And we actually can do this simultaneously over hundreds of neurons. And we did it looking at more than 400 neurons, and specifically these PNOC neurons. So we're doing very specifically to a particular group of neurons in the brain. And we can see that we're getting, for example, 32% of the neurons getting excited, whereas 23% are getting inhibited. But the one key thing, as you're seeing here, is that they're correlating with the arousal response. So we ran further analysis to really show that the activity of these individual neurons is actually correlating very well to the arousal response when they're exposed to an anxiety-producing odor. Another thing we can do is we can use optogenetics. So without going into much detail about optogenetics, what that allows us to do is to go particularly into the brain, and in particular to these PNOC neurons, and be able to modulate them directly. So what we can do is within second time scale, we can actually go into the brain and activate the activity or have these neurons become active. So what we did is, this is a similar experiment where the animal's also head fixed, but what we did is we put a pulse oximeter, which is going to measure heart rate. We have the same camera as what I showed you before. And also in the bottom, we have a little movement sensor that allows us to see if the animal's moving whenever we activate these neurons. And what we saw is that activating these neurons does not cause the animal to move, but it does increase the size of the pupil when they get activated, as well as like the heart rate. So we get this arousal response when you activate these neurons. And then with that, one of the things we wanted to study next is, okay, so we have these neurons that we're able to reliably induce an arousal response. Then can we also look at the activity of these neurons while the animal's freely moving? So while the animal's going to different environments, and in particularly like more like anxiety-inducing environments. So in order to do this, what we do is we use a very classical assay called the elevated plus maze. And the elevated plus maze is very suitable to study how an animal transitions from, as you can see here, a closed arm versus an open arm. You may wonder, why is this anxiety inducing? Well, actually, mice and all rodents don't like to be in open spaces because evolutionarily speaking, they have been afraid of potential predators that could grab them. So this is a really good way for us to study in animals what happens when they go into anxiety-inducing environments. You may also be wondering, but how can we record the activity of these neurons in a similar fashion as you did, as I showed you before, with the 2-photon microscopes? The animals had to be head fixed. So actually, technological advances in our field now allow us to do the same thing I showed you before, but in freely moving animals, using a miniature microscope that we can implant on top of mice and really visualize the activity of neurons while the animal freely moves and explores an environment. So what I'm showing you here is recording activity from the same PNOC neurons I showed you before, but as a mouse explores the closed and open arms of an elevated plus maze. So to go briefly through some data, what you're seeing here is just a pictograph right now of like open arms and closed arms as I was showing you before. But here we can track, as the animal moves through the elevated plus maze, we can track the activity of single neurons and how they respond when the animal, for example, here's a neuron that got excited when they went into the open arm, and a neuron that doesn't get excited when they go into the open arms. And in particular, I can also show you the same data of a neuron being excited, but here, based on where the animal is located, open arms or closed arms, you can see increased activity in the open arms versus the closed arms. And in particular, it's like ramping up as the animal goes to the edge of the open arms. And it was very striking that, similar to what I showed you before, we also get neurons that are excited or inhibited when animals go into the open arms. So these anxiogenic or anxiety-inducing environments, similar to what before we were showing the head fix scenario, that we were showing a odor that induces an anxiety state in an animal. Therefore, one of the things that we found is that these PNOC neurons signal anxiety-inducing environments. Now, one thing we can do is we can go to that optogenetic experiment again and now modulate neurons. So what I showed you before, that when you activate these neurons, you're able to induce an arousal state. But what we've also found is that when you activate these neurons, you actually also prevent them from going to these open arms, into this anxiety-inducing environments. So these neurons are not only arousal-inducing, they're also preventing behavior from animals going into open arms. However, which just takes us to conclude that driving activity increases this anxiety-like behavior. Furthermore, if you do the same experiment, but you now photoinhibit, that you inhibit the activity of these neurons, now the animal is freely moving during both open and closed arm as if they were not afraid of the open arms anymore. So what we show with this is that inhibiting activity decreases anxiety-like behavior. With that, I'd like to conclude of some of the things that I was showing you was that animal models allow us to target and study specific group of neurons that regulate stress and anxiety. Furthermore, that these PNOC neurons, they drive arousal responses. And furthermore, they also signal anxiety-inducing environments. So perhaps pharmacological adjuncts that target these specific group of neurons may help reduce hyperarousal in patients suffering from anxiety disorders. And one thing that I really like to highlight is that there's lots of unique neurons in the brain and lots of work to do. So we definitely would love to recruit many of you to the research field, and specifically as we do animal models to study aspects that are related to psychiatric disorders. And speaking of that, one last thing I'd like to mention is that what we're building currently at the Department of Psychiatry, we're really interested in translational research. So how can we take our findings from the animal work into humans? And with that, for example, my lab collaborates a lot with the lab of Anthony Zanas to do human research work and really take our findings from animal models to potential treatments in humans. And with that, we've been the co-director of the Carolina Stress Initiative together with my colleague, Anthony Zanas. With that, I'd like to wrap up and really just thank the team behind this. This is a picture of my lab and some of the members that did the actual work that I was just showing you today, that collaborate with me every day. And with that, I'd like to thank you all for your time and really now just present to you and pass over the mic to my colleague, Dr. Crystal Schiller.
All right, thanks, Jose. Let's see here if I can share my screen. All right. I'm just going to start talking and assume you can all see it, and someone stop me if you can't.
It's all good.
Okay, great. So thank you so much. I'm happy to be here today to talk about the impact of stress on the brain and innovative treatment. So I'm going to build on some of what Jose just told you about our preclinical models of stress and anxiety and bring that into humans. Before I get started, though, I want to acknowledge that this is team science and thank the participants who contribute to this research because they really put in a ton of effort. And thank my mentors, collaborators, trainees, and funding agencies, of which there have been many, thankfully. And so you'll see throughout this talk that there's a lot of opportunity to get involved in research in the Department of Psychiatry and with my team specifically. So we know, based on what you just heard, that stress impacts all of our biological systems, including the central nervous system. But chronic stress in particular leads to exhaustion, inflammation, depression, and anxiety. And that's no news to you because you've been experiencing that for the last year and a half. So what we've been looking at in my lab is how depression in particular, and anxiety as well, can exert impact on the brain, and how we can see that in real time using functional MRI. So in this study, we recruited women who are currently depressed, and we looked at how they reacted to winning rewards in the scanner. So we had them do this kind of boring task, but basically they had to push a button every time they saw a particular thing come up on the screen. And if they did it fast enough, they could win money. And so as they're lying there in the scanner, we're looking at how their brain function changes as a result of winning money. And what we found is in the group of people with depression, their brain circuits, in particular, the neural reward circuitry, is actually less responsive to winning rewards. And in particular, it was this key node of the reward system, the putamen, that seemed to be less active in people who are depressed. Now, the interesting thing is that then we treated their depression. In this case, we used a hormonal treatment, transdermal estradiol, which is a form of estrogen that women going through menopause experience a reduction in this hormone. And so we use this hormone to actually reverse the effects of depression. And you can see it increased activation of that same area that was reduced when the women were depressed. And at post-treatment, our group of women who were depressed at the beginning showed an increase in this area. Similar by post-treatment, they looked exactly the same as the group of women who were not depressed, our control group. So we know that we can, not only does depression show up in the brain, we can also fix it essentially. So you may be wondering what kind of treatments actually work for this. And as alluded to in my last two slides, this neural reward circuitry is what is dysfunctional in depression. And that's the thing that produces motivation in people. So the motivation to do things, to get up in the morning, to do all sorts of things, but especially to pursue treatment. So it's hard to get people who are depressed into treatment because of the lack of motivation at times. And there's a specific kind of treatment called behavioral activation that actually targets motivation. So the whole point of this behavioral activation treatment is to increase motivation. And the interesting thing to me as a psychologist is that it works just as well as antidepressant medication, even for severe depression. So this was a study done quite some time ago now by my colleague Sona Demijan that showed behavioral activation produced remission or treatment response in about 76% of patients with severe depression. And that was over the course of 16 weeks, and it looked just as good as medication. So there are a lot of barriers, as we talked about earlier, to getting into high quality evidence-based therapy. And so that brings us to a current study that I'm working on in collaboration with Dr. Meltzer-Brody and many others, called Scaling Up Psychological Treatments for Perinatal Populations, or SUMMIT. And in this study, we're looking at the use of non-specialist providers and telemedicine in a randomized controlled trial. It's really interesting. We have 24 investigators, collaborators, and advisors from around the world and 18 stakeholders. It's funded by PCORI, and they've given us over $13 million to do this trial. It's the largest psychotherapy trial to date for perinatal populations. And we're seeing patients in 3 cities, Toronto, Chicago, and Chapel Hill. So the study aims to improve access to mental health care for pregnant women and new mothers, and we're investigating different ways of providing talk therapy to reduce barriers and increase access. And our hypothesis really is that nurses or non-specialist providers are just as effective at delivering behavioral activation as specialist providers, so psychologists and social workers in my program. And we found that, so the rationale for this is that talk therapies are really preferred by women, especially during pregnancy, over medication. But only about 1 in 5 women are actually treated with talk therapy because of all the barriers. So the pandemic has given us another barrier, and in-person, face-to-face therapy is just not possible in the way that it was pre-pandemic. So that's the other thing that we were looking at in this study, is whether telemedicine or psychotherapy delivered via the internet, just like I'm coming to you now, is as effective as in-person, face-to-face therapy. So we actually launched this study before the pandemic in January 2020, and then we all know what happened after that, which gave us enormous barriers to actually doing the work. But I'm proud to say that we've persisted. It looks like I'm missing something. Oh, here we go. So there were some modifications to the study design that happened during COVID-19. In the beginning, so we had 4 study arms. We were looking at telemedicine versus in-person treatment delivered by nurses or specialist providers, like I said, psychologists or social workers. And then when the pandemic really struck, soon after we started, we moved to telemedicine only for a period of time. And recently we switched back and are now doing in-person as well. And it's been really interesting and challenging to ride this out over time. It has required constant pivoting, as well as a whole troop of people to help us manage this. But I am excited to report that we are on track with all of our recruitment goals. We have enrolled over 600 women to date, and over 400 have already completed treatment with us. So you may be wondering, so what's so great about behavioral activation therapy, or how does it work? And behavioral activation is pretty simple, actually. It works by helping people engage in their lives in 3 domains. So we teach people the importance of seeking pleasure and how to do it day to day, even in small ways. So like this woman in the picture, even listening to your favorite song can make you feel happy and actually reduce your risk for depression and anxiety. We teach people about the importance of making connections, which has been super difficult during the pandemic, but so much more important. So how to capitalize on those social connections, engage your social network, not on social media, but face-to-face whenever possible, via text or phone, and how to make those connections meaningful and in ways that matter. And then also, how to celebrate accomplishments. That's not something that we're very good at as a society. We accomplish something and then we move on to the next thing. But the goal here is to slow down and really celebrate our successes. So some of our patients with severe depression even have trouble getting out of bed or brushing their teeth in the morning. And so we celebrate those accomplishments together. And so it's just building on successes over time. All right, so beyond that study, the pandemic has really changed how we provide therapy. Most of our treatments, at least in our clinic and in most places, are actually delivered virtually now. So at the beginning of the pandemic, I was working with one of our clinic managers, and he said to me, "But do these virtual treatments actually work?" And I thought, good question, let's figure it out. So we embarked upon a study to try and understand the efficacy of existing virtual treatments. And I have good news. So first, virtual treatment works about 10 times better than nothing. So depression, if you wait long enough, often gets better, because depression by its very nature is episodic. It gets worse and it gets better. But if you get virtual treatment, so whether it's online CBT, cognitive behavioral therapy delivered by an app, or talking with me or another therapist online, you'll be 10 times better off than if you do nothing. So another option that a lot of people use is regularly checking in with their family physician or other kind of doctor, maybe an OBGYN if you're pregnant. And virtual treatments are actually 2 times better than that. in terms of producing remission and treatment response. We also found that virtual treatment, surprisingly to me as a psychologist, works just as well as face-to-face treatment. So you can treat your depression with an app just about as well as you can do by meeting with a therapist every week. And the reason that that's really good news is that it's really difficult to afford and access high quality psychotherapy. But this puts psychotherapy into the hands of everyone with a smartphone, which is really fantastic. So if you're looking for a virtual treatment, which ones work best? We found that virtual treatments based on evidence-based interventions for depression, things like behavioral activation or cognitive behavior therapy, work better than mindfulness or relaxation-based treatments. We also found that virtual treatments involving check-ins with an actual person, whether it's done via phone, email, or a text, work better than those that are completely unguided. So remember, I said that depression is associated with poor motivation. And I think that that's why these check-ins are so important, because it's a way of ensuring accountability to actually engaging with the treatment. So, but you could do that on your own if you had a support person that you identify, like a parent or a friend, to check in on you as you go through your online treatment. All right, so in sum, we know that chronic stress increases risk for depression, and therapy is both effective and accessible online. Depression reduces the brain's responsiveness to rewards, which actually can be corrected with treatment. The pandemic has caused lots of changes in the way that treatments are delivered, but we know that virtual treatments work just as well as face-to-face treatment, and they're now widely accessible. So with that, I'm going to turn it back over to Dr. Meltzer-Brody, who has some announcements about next week's Mental Health Summit.
Well, thank you. Thank you, Dr. Schiller and Dr. Rodriguez-Romaguera for great talks. And we're going to be able to do some Q&A in a minute. I want to give a plug. The Chancellor has convened a mental health summit, which is a week from today. It's a day-long program on Monday, November 15th. And this is an acknowledgement of the massive mental health impact of the pandemic on the entire world. I presented world data, and the campus is a microcosm of that. And it's really important we come together as a community to educate, engage, and foster discussion. This is for faculty, staff, and students. You've seen campus communications. There'll be more coming back. It's free. You can pop into any part of it. You register online. You have to have a UNC email to be able to attend. And there'll be sessions on student voices, diverse student voices, faculty and staff voices, parent voices, looking at culture. A foundation presentation from the JED Foundation, which focuses on mental health on campuses and how keeping a campus safe to reduce suicides. And then working group presentations looking at prevention, crisis response, and campus culture. And all of these discussions will help us distill a set of recommended action items as we try and address this all together. So hoping many of you will participate and listen in and engage. And certainly anyone needing help, these are the resources for mental health services on campus, and these have been widely distributed, but please reach out and get help if you're struggling at all. So I want to turn to the questions that came in and just say, this is such a profound time, and the awareness raising of the pandemic around the cracks in the system and the increased prevalence of mental health concerns. What you've heard us talk about today are some examples. One of the first questions was, is perinatal depression or postpartum depression different than other types of depression? And there are some differences and similarities for sure. And the underlying biology is an area of great interest, and one that would be a much longer conversation to have. We certainly know that there's probably genetic differences, and there can be some differences in presentation between a major depressive episode that occurs outside of the perinatal period and one that occurs during. But I want to go back to some questions that people asked earlier. There were some questions to Dr. Rodriguez-Romaguera. including pharmacologic or clinical approaches that could potentially be implemented to suppress neuronal activity. And that was a question that came in. So Jose wondered if you wanted to take a stab at that. It looked like the first question that came in for you, you then answered in your talk. But I thought that question was a great one for you to answer.
Yeah, thank you, Dr. Meltzer-Brody. So yeah, so the question, as I understand it, is, are there any pharmacological ways that we can modulate PNOC neurons, right? So actually, one of the things that we know, so PNOC is actually the gene or the genetic precursor to the nociceptive neuropeptide. So for example, neurons, when they communicate with each other, they're going to be releasing things. Like one of those things is neuropeptides. So neuropeptide release is going to have an effect on neurons that are receiving information from PNOC neurons. And right now, one thing that we actually have is we have antagonists of that receptor. So we can block the receptor where nociceptin activates. So that could potentially be a way to modulate the output of PNOC neurons. However, we still do not know what receptors exist within PNOC neurons that we can then modulate. So that would be a way to directly modulate the activity of PNOC neurons. But what we have available right now potentially is to modulate the neurons that PNOC neurons has its effect on. So, and that's something that definitely we're very interested in pursuing that could potentially lead to some novel treatments.
Fantastic. No, thank you, Jose. Dr. Rodriguez-Romaguera's lab is doing some very, very cool work. It was really fun to hear about that today. Dr. Schiller, it looks like you have a couple questions coming in here. And one I think you talked about, but let's say that two questions to think about. Does exercise have a similar influence on depression to that caused by behavioral activation? We'll start with that one. And then recommendations on specific CBT virtual treatment apps that you would recommend.
Yeah, that's a great question. So exercise is very commonly a part of behavioral activation as implemented in our clinical practice. I think almost all of my patients who have gotten behavioral activation set an exercise goal. It does seem to elevate mood, to move your body. But behavioral activation goes much further beyond that to help people in other domains of life as well. So I do think that exercise does have an important mood regulatory effect. It's not the only piece of the puzzle. And for some people, it may be enough. You know, if you don't have really severe depression, I think exercise is a great place to start, especially after you talk to your physician to make sure it's safe, whatever you're planning to do. But yeah, I think for people with more severe manifestations of depression, they need a little bit more than exercise. Was there something else?
Yes.
Specific apps. Yeah, I'm not really sure that I would recommend any app in particular, but I think it's important to look for one that was developed by a psychologist or psychiatrist, a physician of some kind. There's a ton of startups in this space trying to develop new and innovative things that aren't necessarily evidence-based. So you want to see that it has cognitive behavioral therapy or behavior activation therapy, acceptance and commitment therapy. Those are some buzzwords that you'd want to see attached to an app like that.
So data-driven is always good. We always want things that actually have data to support what they're doing. I see another question here for Dr. Jose Rodriguez-Romaguera. The question was, do the cameras tracking the excitement of neurons require the top of the mouse's skull to be tampered with or removed? And how exactly do the cameras work? Great question.
Yeah, great question indeed. So we actually have to do surgeries in the mice where we have to do what we call intracranial surgeries, where we actually implant a lens into that goes in right above the region so we can visualize it. So the best way to think about it is we're doing microscopy techniques, but by putting what I like to call a relay lens that goes directly into the brain region of interest. So we're basically doing microscopy, but at a very high resolution and at a very high time frame, so we can actually image the activity of the neurons in real time. And the cameras, as I said, it's basically microscopy. And yeah, and I hope that answers your question.
Terrific. Great. All right. And then I think that this is a great question I'm going to throw out for both Dr. Rodriguez-Romaguera and Dr. Schiller. Do we work with undergraduates in our labs? And what advice would you give to undergraduates for getting involved in this kind of work?
I can start and then I'll turn it over to you. And maybe we can comment on what undergraduates in our labs do.
That would be great.
Kind of specifically. So yes, undergraduate students work in my lab, mostly through the work study mechanism, but also as volunteers and paid employees. And the first step is just reaching out to me via email to see what opportunities we have available. We have a number of different NIH-funded studies going on, and undergraduates in the lab assist with all aspects of the research. So because most of our data from subjects or research participants is collected via online portals, you know, gone are the days where undergraduate research assistants would sit and type transcriptions of questionnaire data into computers. Now our research participants do a lot of screening of potential research participants. So our undergraduates are trained to screen for depression and answer phone calls to the lab for people wanting to participate in our studies. They also help us collect brain imaging data. So they're trained to administer brain imaging tasks at the scanner, help us scan subjects' brains, help us do quality assessments of the results and analyze data. Majority of the undergraduate students in my lab have gone on to do honors theses. And so they may do original data collection by identifying a research question that they're particularly interested in, and I will help them design a study and collect data. Or they may use existing data from our lab to ask a new question and see it all the way through from hypothesis testing to data analysis and then writing up the results for publication. Jose, do you want to go next?
Yeah, and that's very, I love the enthusiasm from the undergrad community to get involved in research. And one of the advice I would give to any undergrad that's interested in research is the one thing is to go on our websites, go on the websites of the different faculty that are in the psychiatry department, for example. Look at what people do. A lot of the times our websites describe what we do. And sometimes even we have like articles in there of the actual research we've done. So if I could provide the best of advice is to find out what labs do, read up on it, and then see if part of that excites you. And then when you write us to contact us, tell us what excited you. Tell us like, oh, I was able to understand some aspect, doesn't have to be the whole thing, of one of your articles, and I found this to be very interesting, and that to be very interesting. So that type of email is the best type because it's the one that's telling us like, oh, you know, you, the human being is interested in the questions we're pursuing. So then, you know, it would be great if you joined us so we can tackle some of these problems together. And along the other way of like what students do, for example, in my lab. So in my lab, students will many times run behavioral experiments. For example, the behavioral experiments I showed you, putting animals in elevated plus maze, tracking the activity of the animals moving around. And furthermore, for example, they also do like histological analysis. So like looking at actual brain tissue and looking at like how these cells are labeled and what are they co-labeled with, what are their markers they're labeled. So a lot of the work that we do, you know, it's what an undergrad gets involved with, it's up to them how fast they want to go. We can take them all the way from doing behavioral experiments to doing surgeries. So if that's what they're interested in and they want to work towards that.
Well, fantastic. Well, I love the questions, and I would say I've been around for decades here, and I've had the experience of working with undergrads that came to grad school and who then went on to be faculty here or elsewhere in a variety of different things. And that's just amazing to see. So please, we love undergrads getting involved. I'll end by saying, thank you for coming and attending this today. Make sure you're looking out for yourself. Stress and mental health concerns right now are impacting all of us in different ways. Do not be shy about reaching out. You're in very good company, and there's a whole lot that can be done. And it's an amazingly exciting time in neuroscience, in psychiatry, psychology, and behavioral health to get involved. So the next generation is going to make a major splash, and we're here to help in whatever way we can. So be well. Thank you for coming today, and thank yous for Dr. Rodriguez-Romaguera and Dr. Schiller for the terrific talks. It's been a pleasure to spend time with you today.
Thank you.
Likewise.
Take care.
Everyone.
Bye. Thanks.